All right, people are joining. I want to thank everybody for joining, and we'll get started in just a minute. I think we'll give everybody a minute to join. Thank you to everybody for joining. Allison, one thing I actually can't see because of the way my screen is set up is sort of when, as people are joining, sort of when that slows down so that so that we can sort of think that people are maybe here.
I think we're okay to start now, Dr. Berry.
Okay, terrific. Thanks. Well, I want to thank everybody for being here. This is really exciting. These webinars have been a lot of fun to present at over the years, and I'm excited that we're really dedicating, one of these a month to our expanded access programs. Today, I'm going to talk about the RAPA Therapeutics Expanded Access Program, and this is of something called epigenetically reprogrammed T stem cell therapy, and hopefully that will become at least sort of clear about what we're talking about. It is a T cell therapy, so it's a cell therapy, uses T cells, which are a kind of immune cell, and they are reprogrammed. And what we mean by epigenetically really is just there's no genetic manipulation. They are not genetically modified, so they're the person's own cells. So we'll walk through all of that. I should say, actually, before I go on, I should say that this is a program that I'm running in conjunction with Drs. Babu and Paganoni, as well as in very, very close collaboration with RAPA Therapeutics, who's the regulatory sponsor. The program is funded by NIH, specifically the National Institute of Neurologic Disorders and Stroke. And one component of this is home monitoring using digital tools, and we've partnered with Everything ALS for those tools. And so that really been a great collaboration. Some complexity behind the scenes, and I think that means we're going to get great data. So I want to start with just sort of giving you an overview of this cell product, which is, in this case, the therapy that we're talking about. So RAPA cell therapy, actually, really, the development goes back to the NIH, to the early 2000s and mid 2000s, when there was a lot of work run by Dr. Dan Fowler, who was the chief scientific officer for RAPA, was at the NIH at the time, and then really in 2017 spun the company RAPA out of some of the work that he had done there. One of the foci of RAPA Therapeutics, and a really big one, is actually oncology. And so they're developing cancer cells for cancer therapy. At the same time, they've been developing, since 2018, cell therapy for people living with ALS. And that started with a single, what was called at the time, compassionate use or expanded access program, single patient. And that really, I think, gave them the confidence to build a phase 1/2 trial. And we were a part of that trial. There was another site in New Jersey. We treated scores of patients, actually, with varying doses of these cells, with and without chemotherapy as a sort of pre-treatment. And really came to a place where we could develop a phase 2/3 trial that was much simpler, and expanded that into this EAP and applied for funding. This is after learning a lot in the phase 1/2 trial, this is a really streamlined therapy, and one that we thought we could bring successfully to EAP, and you'll see that we're doing that. So RAPA-501 are what are called autologous cells. That means they're derived from the person that we're treating. So the same person that is a participant in the study will give blood in a special way. We take white blood cells, that is the immune cells. They're harvested through a process called apheresis. Apheresis is kind of, I call it the Jiffy Lube of blood draws. So you have really sort of one IV in one spot that takes blood out, puts it through a machine that keeps the white blood cells and puts the rest of the blood back in through a different IV. And it takes a couple of hours, and that harvests enough immune cells that we can then expand those for preparing the RAPA-501 therapy. It does not take enough cells that the person is left immune depleted or unable to fight off infection. It's not a huge sort of number of cells, probably equivalent to what you might take in a couple of pints of blood, but the rest of it goes back. We just hold on to the white cells. The second step is that those cells are then shipped from the hospital where they're obtained to the manufacturing facility at RAPA. And the white blood cells are separated into different subsets, and the T cells are kept. So T cells are one of the immune cell types. There are others, monocytes, B cells, natural killer cells. There's a whole lot of different cells, but we hang on to the T cells. And those T cells are then de-differentiated. That is, they're grown in a condition that makes them less specific. So there are lots of different subtypes of T cells, and they're grown in a way that makes them all sort of naive T cells. And that's a starting point for then what's called redifferentiation, where these T cells are turned into a kind of a cell that is actually not exactly any one kind of T cell, but a very useful cell for us. So it has some characteristics of what's called a regulatory T cell and some characteristics of what's called a Th2 cell. And I'm going to talk a little bit more about that in just a moment. After those cells are created or made, they're put into sterile bags, they're frozen, and then they're shipped back to the hospital. And then the participant comes in, and I'll show you our schedule for this, but comes in for infusions of those through an IV infusion. And that's the treatment that we're talking about. So again, I'll go into some of the details in just a minute. We have to unravel this ball of yarn a little bit at a time. So I want to talk a little bit about what these RAPA-501 T cells are doing, and then I think you'll begin to understand in the next few slides sort of, how they're made and why it's important that they're made that way. So we'll walk through these steps 1, 2, 3, 4, 5. In the first step, in ALS, the motor neurons are becoming ill, and as a part of becoming ill, they and the cells around them begin to secrete these inflammatory factors. We call them cytokines or chemokines. They're pro-inflammation. And they're molecules, and they spread throughout the blood and the body. And that inflammation can actually damage surrounding motor neurons and can, we think, be a part of hastening the progression of the disease and making other motor neurons ill, and in fact, making that same motor neuron more ill. When we put the RAPA-501 cells in, those RAPA-501 cells are sort of engineered to be this hybrid cell. And what happens is when they encounter these pro-inflammatory factors, it actually activates these RAPA-501 cells. And the RAPA-501 cells, when they're activated, become anti-inflammatory. So this same inflammation that's sort of activating the immune system is activating our RAPA-501 cells. And then the RAPA-501 cells actually decrease inflammation from other T cells in the body and decrease that inflammatory cycle. So they kind of take that pro-inflammatory signal and they turn it around on its head. And that's why we have to engineer them to be sort of parts of different kinds of T cells. And I'll talk about that on this slide. I'm not going to go into some of the details like telling you about what mTORC1 and mTORC2 blockade are. But essentially, when we create these RAPA-501 cells, what we're doing is taking away the fate that would make them pro-inflammatory cells. So that an activation of these cells does not make them pro-inflammatory because they don't have the mechanism to become pro-inflammatory. They get activated, but they don't become pro-inflammatory. Another thing that happens with these hybrid cells are called Treg Th2 cells. Another thing that happens with these cells is they're grown in a very, very hostile environment for part of the process. And the weaker cells don't make it through that. So it's selecting for cells that are really metabolically fit. They're ones that will remain T cells, they'll remain active, they'll remain anti-inflammatory when they're put back into the body. And that's really important because if we don't go through that step, there's a possibility that you can put regular old regulatory T cells into the body and they can actually switch and become killer T cells in the wrong circumstance. So we want to avoid that, and that's part of the process as well. The other thing is that some T cells can become very short-lived. And so there's a part of this process that removes checkpoints for T cells. And those checkpoints are kind of sort of, if you reach these checkpoints in the life cycle of a cell, then the cell begins to self-destruct. And by removing those, these cells live longer. So they're sort of being goosed along the process to become non-inflammatory, but very strong, long-living. And then the last piece of this down in the lower left-hand corner is that they have multifaceted immune suppressive function. So they have regulatory T cell properties. Regulatory T cells are actually cells in our body that sort of tamp down the immune system, and they have some of that. There is a related kind of cell called Th2 that has some inflammatory and some anti-inflammatory properties, and they retain the anti-inflammatory properties of those. They also retain the ability to be activated by the immune system. And they even have some of the homing molecules on them that allow them to go to sites of inflammation in the body. So it's really kind of sort of moving these cells along to reprogram them to be some pieces of all these different kinds of cells so that they have the best chance of being anti-inflammatory, being in the right spot, being long-lived, and not turning into kind of a bad guy, kind of a T cell. This slide just sort of shows that they express two really important factors. One is called FOXP3, and this is a factor that identifies a cell as a regulatory T cell, and regulatory T cells are anti-inflammatory. But they also express some of the factors that make a Th2 cell, which is another, this mixed kind of pro-inflammatory, anti-inflammatory cell, and that's called Gata3. And so they're not a pure Treg population. They're not a pure Th2 population. It's really just showing that they are this hybrid population that we're aiming to create. And then, finally, and I'll come away from some of the really complex stuff after this, but they do also express something called CD150, which is a T stem. It's sort of expressed on T stem cells, that is very naive stem cells that are T cells. They're not really stem cells, they're T cells, but they're sort of naive T cells. And that means that they have the flexibility that's built into these young T cells. And again, that was part of the process that I showed you of kind of creating these naive T cells. But this is just showing that they express these markers. So there has been work done by RAPA to show that these RAPA-501 cells, these sort of hybrid cells, do suppress CNS inflammatory microglial cells through co-culture. So in Petri dishes, essentially you culture together inflammatory CNS cells along with the RAPA cells, and you can see that there's less inflammation when you culture them together than when you just have the inflammatory CNS cells. And that's, I think, a good proof of concept to say, these cells are actually doing what they should do. Having said that, it is in a Petri dish, and so it's important for us to go beyond that. And that's why we did the phase 1 trial. It's one of the many reasons we did a phase 1 trial. The other is safety. And by the way, we saw a very good safety profile. But we were able to look, and if you look on the left side, you can see one participant who's sort of pro-inflammatory profile was really elevated pre-therapy, and you can see how it comes down dramatically. And this is on a log scale, so it's actually even much more dramatic than it looks on this graph, but really brings down that pro-inflammatory profile. And then on the right side, you can see a number of different participants who show varying degrees of reduction. So you can see the 99% reduction. Not everybody got to that level, but there was a substantial reduction in the inflammatory profile after treatment and these participants in the phase 1 trial. So not only did we find that it was safe, we did find that we were engaging the immune system the way that we had hoped to do. And this work really comes out of very careful work that is done at RAPA Therapeutics. Again, all of that science coming out of the NIH. So I want to talk a little bit about how we're now incorporating that into the EAP. And I think the first thing is to say that this is really, this EAP is funded by the NIH through the NINDS. This funding comes from the Act for ALS money that was designated through a bill that really came from the community. And it was really that community showing our government, the people who hold the purse strings, that this is really going to impact people's lives. And that's how we got to where we are. Now, the NIH, and I think you've probably heard this on other webinars, but the NIH is very excited to be supporting these EAPs, but also says, look, this is an opportunity for us to do two things at once. We can provide access to experimental therapies, and at the same time, we can answer some questions about science. And I think this program is at a uniquely is uniquely positioned to really learn a lot from this EAP for a variety of reasons. But one is that there's a lot to be learned about cell therapy. Any opportunity to treat a person with cell therapy is an opportunity to learn. Any opportunity to look at the immune system from a person is a real opportunity to learn. And it's such a rich environment to look at biomarkers and look at targeted effects. The other thing is that we don't have a lot of experience treating a huge number of people with this therapy. And so we're learning from every person that we treat about safety and about what its effect is. And then finally, we really have positioned this to treat a new population of people, that is, those who are later in the disease than those who were treated in the phase 1 or who will be treated in the phase 2, 3. And I think that's really important, particularly because we're targeting the immune system. And we do think the immune system changes over the course of the disease, such that when people are further along in the disease, the immune system does seem to look different than it does early in the disease. So I think it's really important that we see, okay, we know that when we targeted people a little earlier in the disease, that we could make changes in the immune system. Can we do that when we target people later in the disease too? So we're answering some really critical questions. And for that reason, this is being run just like a trial. All the bells and whistles, all the safety monitoring, all the biomarker monitoring, and more. But it just is open label. We don't have a placebo. And I'll tell you what we're doing to sort of try to get good data even without a placebo. I think I talked about the fact that this is, NIH funded, that the grant program came out of the Act for ALS, and that we're really specifically making this for people who aren't eligible for ALS-related clinical trials. The mantra about expanded access programs is that they shouldn't interfere with ongoing clinical development. We don't want to do EAPs at the expense of trials. We want them to add to a clinical development program. And I think we positioned this well this way. So one of the key inclusion criteria for this is that the vital capacity should be less than 50% of predicted normal. And that differentiates this population completely from those who are in the phase 1 and those who will be in the phase 2, 3. It does two things. It does, as I said, it makes it so that it won't interfere with clinical development of RAPA-501, but it does really get us experience in an important, different population, I think for a lot of good reasons that I outlined. Other than that, we've really tried to be, hew to this idea that expanded access programs should be open, and so it can be sporadic or familial ALS, any LS-Goriel criteria for those of you who are familiar. So it can be, have spread a lot or not a lot. People can be on other medications. There are no restrictions from the time since diagnosis. You can't be in another EAP. You can't be in another trial. I mean, that's sort of a kind of, we couldn't sort of find a way around that. But otherwise, it's pretty broad. The one other note is that people do have to have enough T cells at baseline for us to get enough cells to manufacture. And so that's just, we anticipate that most people will meet the criteria that we've laid out, but there may be some people now and again who don't have enough T cells. We do see a little bit of immune dysfunction in people with ALS. So I wouldn't be shocked if we run into someone here or there who fails screening for that reason. We're looking for 40 participants. And again, these are people who won't be in the clinical trial. We want to accrue people quickly, and we want a diverse population of people living with ALS. We want people from all walks of life, from all areas, from all races, from all ages, to be a part of this study. That's important to us. And I think saying it out loud and saying that we are here to include people in this broadly is really important. We're going to collect data about safety and tolerability. Are there side effects? I will say we have not seen side effects. One of the potential side effects that we look for is an immune reaction or sort of like an allergy to the cells. Remember, these are the person's own cells, so we don't think it would be a reaction to the cells, but rather just the fluid that the cells are packaged in. We're always on the lookout. We haven't seen other kind of adverse events or side effects that we think we need to keep an eye on, but we always have our eyes open. We want to look at the effects on immune function. We'll be collecting blood and doing those studies like I've shown you and many more at RAPA Therapeutics. And also we want to look at neurofilament light and see if we can see a difference in neurofilament light over time. We'll be looking at clinical characteristics like the ALS Functional Rating Scale revised, the ROADS, which is another functional scale, as well as vital capacity. And then what I haven't talked about yet, I mentioned in the beginning, but haven't talked about it until now, is everything ALS. So we wanted to really get as rich a data set as we could. And we approached everything ALS, and they've agreed to put together kind of a digital kit, which then goes to the participant at home. Not every participant has to opt into this. They have they can say yes or no to this part, but we think it's an important step forward for gathering data. The kit will include a cell phone, a home pulmonary function machine called a spirometer. On the cell phone will be apps to fill out surveys, to record your voice with oral analytics, and then also a wearable device to look at activity monitoring. So that is all packaged in a way and comes from everything ALS in a way that we hope is easier for the sites, for the site staff, so that they're not fielding a lot of questions, that the experts in this, the people at Everything ALS, will be fielding questions, and easier for the participants because it's all packaged up really nicely, and there will be specific support from Everything ALS to kind of get you started, and a call every 2 weeks to walk you through some of these questions. We'll be doing this at 10 trial centers, and I'll show you those trial centers in just a minute. The primary outcome is really to provide people who are living with ALS with access to RAPA-501. But we also, as sort of a very close second, is to evaluate feasibility and safety. And then I've talked about some of these secondary objectives of characterizing the immune system, looking at neurofilament, looking at clinical measures, and looking at the digital measures, which is an exploratory. And then one other piece to this that I really haven't talked about yet is we're working with a company called Origin Data Sciences, and they have a prediction algorithm so that they can take baseline characteristics and predict how ALS will progress. And that we're going to use that as what we call a synthetic control. Some people call it a digital twin. It's a prediction of how the disease might progress, and then we'll compare that to what we observe, and we'll do that for changes in ALS-FRSR, vital capacity, and survival. Now, I want to be very careful to point out that this is our best attempt to try to gather information about effectiveness in the absence of a placebo group. It is not the same as having a placebo group. So we hope that this will give us some insight. It's not going to answer all the questions, and we're aware of that. So we're just trying to get an edge here. These are the 10 sites that we think will be participating. We don't have all of them up and running yet. There could be changes still, but really thankful for all the work that's going on at these. Providence and Portland, UCSF, UC Irvine, Barrow Neurologic Institute, and Mayo Clinic Phoenix, University of Minnesota, Iowa, Emory, Hackensack, and MGH. All of these sites are making progress in getting up and running. This study does take, building a team that's a little outside of just our ALS team, interacting with the people who do this apheresis with cell labs and so forth. For the kind of flow of this, from the perspective of a participant, if somebody is interested in this, they would call their center, get set up for a screening visit. That screening visit, takes probably 2.5 to 4 hours. And I'll show you in a minute sort of the kinds of things we're doing, but drawing blood, looking at safety, looking at vital capacity, getting medical history, and looking at the vein. It's no small thing, but looking at the veins to see whether we can do an apheresis and get the blood for this. 40 participants will, screen in to the study, and the next time we see you at the site is for the apheresis. So you come into the site for screening, and then within a month to 6 weeks, you come in for this apheresis, which again is sitting in an infusion chair basically for 2 or 3 hours while the white blood cells are harvested. About a month, roughly a month after that, the participant would come back in for the first infusion, and then there would be infusions every 6 weeks, and we do up to 4 infusions. We plan for 4 infusions. It's possible, if the production doesn't go right, that we would have too few, and somebody might only get 3 infusions, for example. But we plan for 4 infusions. Those infusions are actually fairly short. We have to get people settled, get them into their infusion center, do some sort of baseline ALS-FRSR, vital capacity, and so forth. And then the infusion itself lasts for just a matter of minutes. There's a flush that lasts for a few more minutes after that. And then we observe people afterward for a couple hours just to make sure that they're doing okay. After those 4 visits, then there's no more visits to the site, but there will be 2 more remote follow-up visits after that final infusion, just to see how people are doing after the infusion. And through all of this will be the Everything ALS monitoring that's done at home. We've minimized, and this is a big difference from our phase 1 where we had lots of blood draws. We're trying to understand where the cells are going, what their effect is. Every time we had many visits to do that and look at biomarkers. We've really tried to minimize this to just the visits we need to treat people. And we'll be taking blood to monitor biomarkers at those visits. But it's a pretty streamlined flow, which is really exciting that we've gotten to that. So this is, for those of you who think in grids, this is the schedule of activities. So you can see you come in for screening. We do a lot of these sort of, basic demographics, disease history. We do have to test for viruses, and that's a requirement for any cells that are grown in a cell lab, kind of our clinical outcome measures. And then you come in for apheresis. That's within about a month. And then cycles 1, 2, 3, and 4. They're called cycles because I don't know. That's what they call them in oncology, to be totally honest. Each one of those is just an infusion of the cells. And then we have those virtual follow-up visits. I'll pause there. I really want to give a real true thank you, deep gratitude to the NIH for funding this, to my entire team at Mass General, particularly Megan Okoro, who is a clinical research coordinator extraordinaire who's really helped us get up and running, to the entire team at RAPA Therapeutics. Dan Fowler, this is mostly his science, and it's really quite amazing. Jenny Sunga and Sylvia Yip have been just an incredible team getting us organized and off the ground. And we're working with Osmosis Clinical Research Organization. All of the sites have been working to get going. And then Origent and Everything have been great partners in helping us put together what this will look like. We've also received support from the ALS Association and ALS Northwest. And then finally, people who go out on a limb and participate in this kind of trial and the families who are supporting them. It's remarkable, and a huge thank you. So I think I'll take this down.
Thank you, Dr. Berry. I know we're coming close to the end, but may we keep you for a few minutes to get through some questions?
Couple minutes, Rebecca. Yeah.
Okay. Yeah, I loved how you explained everything. Thank you for doing that. It's like there's a game of survivor going on in our cells with this therapy.
That's right. That's
May
right.
the best cell win. Okay, the questions that are coming in, I'll try to summarize them. First one, does this only benefit respiratory or does it benefit other symptoms?
So the hope is that this actually has an effect for motor neurons. And I think then the question for us always as trialists is, where will we detect that we're having a benefit? Where will we detect that we're slowing the disease? And it may be an ALS functional rating scale, but it could be that we see it in respiratory function. My sense is we might see it in respiratory function, but it wouldn't be specifically targeting respiratory function. It may just be that that's where we see a difference. And in fact, we have some hints that may be something that we see, and we're looking closely at respiratory function, but I don't think it's targeted specifically there. I want to be very clear that the reason to have people who have a lower than 50% vital capacity is that it was a clear way for us to delineate people who were later in the disease. There are lots of ways we could have done that. But this makes a clear line between people who are earlier in the disease and are in the trial and these people who are later in the disease in this study, so that we can have clear populations and look at the differential effects on their immune system.
That makes sense. Can you explain briefly the difference between forced vital capacity and slow vital capacity?
Only sort of. Forced, because they're very similar. Forced vital capacity, in its very simplest sense, forced vital capacity cuts off its counting when you reach below a certain flow. Whereas slow vital capacity will continue to count that flow. There are also slight differences in how you actually operationalize this with the machine, when you take tidal breaths, when you don't. So I think actually what we've really found is they correlate very highly. To reduce variability. We choose one and we go with that. And we've
Okay.
sort of settled on slow vital capacity, but they're very, very similar.
Okay, so for those in the study that had a reduction in inflammation, did their ALS symptoms improve? Like, did they get better?
Yeah, so I don't know that I can say that we saw clear improvements in symptoms, but I don't, our phase 1 study wasn't really designed to follow people long enough or enough people to tell whether we were slowing the disease, and I think that's really kind of the main goal here.
Okay, how is inflammation measured?
Well, it's complicated. So we measure it. That's a very good question, and I wish we had better what we call biomarkers. But for everything we want to measure, like inflammation, like neurodegeneration, we need something that we're measuring. And in this case, we can either measure proteins that are in the blood, cytokines, chemokines, these sort of pro-inflammatory signals, or we can measure cells themselves. How many natural killer cells are they? How many regulatory T cells are they? How many killer T cells are there? And we're doing a combination of those things here. One of the challenges with measuring inflammation is that there's a lot of variability day to day. If you have allergies, if you get a cold, there are just lots of things that can influence that. So we're going to have to be able to read through that. And it's a little bit of a challenge, but I think you need a holistic view with lots of measurements to get a good idea.
Great. And there are a few questions, Dr. Berry, about dosing and timing and sites. And if you've already dosed somebody, do you know yet? Can you kind of just explain the process?
Yeah, sure. So first, the only center, the MGH is the first center that's up and running, the only center that's up and running now. We're aiming to get another 3 or 4 sites up and running over the next 2 months or so. All the sites are making good progress toward this. There are a number of steps to that. So we have to get contracts done. We have to get IRB done. There has to be a team that's put together at the site that will, sort of shepherd people through this, oversee all the regulatory aspects. And in this case, we need to work closely with cell labs and with apheresis units. Not every hospital has an experimental cell lab. So then they need to kind of contract with another organization that would help out. So some of the startup stuff is not for the faint of heart, although the sites are making incredible progress. We have actually done our first 2 infusions. So we've treated 2 people. Everything ALS is up and running now with our participants. We have another participant who's awaiting their first infusion, which should be in the next few weeks. And then we have screening going on. But I think the real key here is we want to get more sites up and running as quickly as we can.
And do you have to be a resident of the United States to participate?
There's no inclusion criteria that says you have to be a resident of the United States to participate. The visits, because these are infusions and it's taking blood and it's giving cells back, and all of this has to be done on site.
Perfect. And I just want to remind all of our listeners that this recording will be on the HEALEY website. I know, Dr. Berry, you did provide us with some slides that show what to expect if you're participating, what sites are included, and there are some questions about that. So I just want to let everybody know you will be able to access that hopefully within the next few days. It'll be on our website. So as we close out, Dr. Berry, do you have any final words?
I just want to, first of all, I just want to say what a joy it is to be here and to, to really just be able to get together on these webinars and other webinars, and I just am, it's an amazing community. And I think I'm proud to be a part of it. And, I think I just hats off to everybody who's participating. People are reaching out for opportunities. We know that not everybody, but more people reach out than we're able to accommodate. And we're working very hard to build capacity so that we can, bring in more people who are eagerly wanting to move the ball forward and do something for themselves too. So thank you for being here. Thank you for reaching out to us as sites. And it's an amazing community.
Thank you for joining us tonight. Thank you for all you do for the community. I hope everybody has a good evening. We'll see you next week. Bye-bye.